Children, in this video we will be learning about the CHEMI-OSMOTIC hypothesis that is occurring in the thylakoid of the chloroplast and why this is needed. This phenomena basically helps in synthesizing ATP, that's the higher energy molecule, from ADP. And second thing is, it also helps in balancing the hydrogen ion concentration inside the lumen of the thylakoid and also the outside of the thylakoid, that is stroma. Right, children? This is an important thing because wherever you will be having higher hydrogen ion concentration, it will lead to increase in the acidity, and the lumen of the thylakoid cannot withstand that much higher acidity. So this has to be balanced time to time. Now what happens exactly here? See, again this is the same diagram you just learnt few minutes back. That was the diagram of the thylakoid. It's a double membranous structure. So you see the outer membrane and the inner membrane, very similar to the one that I drew, and then outside will be the stroma of the thylakoid. Stroma, that means surrounding the thylakoid. The stroma is in the chloroplast and inside is the lumen. So this area is lumen, right? You have to always learn this, that these two locations are different and accordingly they have their own specific functions, right? So once you have made it clear that which area is stroma and which area is lumen, these ones are membranes, outer membrane and inner membrane. So you have to specify a few things here. This is photosystem 2. This one is a cytochrome complex associated with, you know very well that during non-cyclic photophosphorylation, the electron, once the light falls on this photosystem, at that time this photosystem gets excited and the electron is released, right? This electron is taken up by the primary acceptor, and this primary acceptor has to transfer it further. Now what happens, children? In the surrounding stroma there are hydrogen ions present, right? So what happens next? This primary acceptor transfers the electron further, not to an electron carrier, rather to a hydrogen carrier. So here the hydrogen carrier will be PQ. You know very well that three things I have always told you, that an electron transport system contains PQ, cytochrome and PC. So these are hydrogen carriers, proton carriers basically, right? So what happens here? Again I am telling you that light falls on photosystem 2. Photosystem gets the reaction center of the photosystem gets excited, electron is released out. This electron is taken up by primary acceptor. Primary acceptor carries it further, transfers it to the hydrogen or proton carrier. This proton carrier here, you know what happens? This PQ, when two hydrogen ions are taken from stroma, it converts into PQH2. So this is what is happening here, but this is very much temporary because you know very well that when the electron is transported from photosystem 2 to photosystem 1, it's always this way. That means depending upon this direction is basically the downhill direction depending upon the redox potential, right? So from high energy to low energy the things are moving, and during this movement of electron transport, the energy is basically utilized by this cytochrome, right? What happens? The electron taken up by plastoquinone. Plastoquinone, what does it do? It transports the electron further, but also helps this cytochrome to act as a pump, right? Because these are friends, plastoquinone and cytochrome, these are associated, and plastoquinone is a proton acceptor, proton carrier. So two things are happening simultaneously here when this electron is transferred. One thing is going on as electron transfer from PS2 to PS1, simply the way you learnt in non-cyclic phosphorylation, but at the same time during this transport of electron, the energy moving downhill, the energy is taken up by cytochrome, and cytochrome accepts two hydrogen from outside and pumps it into the lumen. Two hydrogen ions are pumped every time this transport is done. The hydrogen ions are pumped into the lumen. So this is becoming quite confusing, isn't it? I mean, the electron is released by photosystem, taken up by primary acceptor, and then primary acceptor transfers it to the proton carrier rather than electron carrier. But then proton carrier, since it has a kind of a mutual understanding with the cytochrome, the electron carrier, so it further transfers the electron to cytochrome only, but meanwhile it also takes out this conversion. It pumps the hydrogen after making a deal with cytochrome. This plastoquinone pumps hydrogen from outer side, that is stroma, right, from outer side into the lumen of the thylakoid. So ultimately what is happening here? Here the lumen of the thylakoid is basically receiving much hydrogen ions from the stroma. This is one thing. Another thing is, you know very well, you have learnt it in the previous video, that here you have the water splitting system, the complex which breaks off water molecule into hydrogen, 2 hydrogen plus 2 electrons, and then oxygen atom. So when you balance it, it becomes like 2 hydrogen, you balance it and 4 hydrogen ions will be there and 4 electrons will be there, and oxygen will be released as the byproduct. So you are again concluding a phenomena that oxygen, which is released as a byproduct during photolysis of water, this phenomena is actually occurring inside the thylakoid, the lumen of the thylakoid, right? So as of now what you can see is electrons given to photosystem 2, oxygen diffused out as free oxygen. So now the remaining content is hydrogen. So now children, you have to see that hydrogen is actually accumulating inside this lumen of the thylakoid from two different sources. One which is pumped from the stroma into the lumen by cytochrome, and second one during the photolysis of water, and ultimately what is happening? The hydrogen is becoming higher in concentration inside the lumen, and this thing is basically causing high concentration of hydrogen inside the lumen and low concentration of hydrogen in the stroma. So you may call it as the proton gradient or concentration gradient phenomena. So therefore hydrogen has to be released out in order to balance the concentration inside the lumen. How this hydrogen will be taken out? This will be given out with this ATP synthase. I have already told you about this ATP synthase. It has two complexes, two structures, F0 and F1. F0 is basically a channel protein embedded in the membrane, and F1 is acting as the enzyme ATP synthase. When hydrogen through this F0 and F1 complex comes out, it is given out as facilitated diffusion, right, through channel protein. This hydrogen is released into the stroma, and at the same time, because things are moving from high concentration to low, the energy is released, and that energy released, I am saying released, not used. So that energy moving down the concentration gradient, energy is utilized to convert ADP to ATP, right? So whatever was happening here, we will be taking a recap at the end. The last thing here that you have to understand is that when this electron is transported to photosystem 1, the reduction of NADP to NADPH takes place. Now you know already that here is the hero of this reduction process. So here is sitting NADP reductase. This enzyme will be carrying out the reduction process to convert NADP to NADPH. You will be wondering from where this H is coming. Just now you have thrusted so many hydrogen ions from lumen. These hydrogen ions will be used by NADP. When it gets reduced by using the hydrogen ions from the stroma, it converts into NADPH2. Right? This is how the hydrogen ion concentration inside the lumen of the thylakoid is balanced, and associated phenomena is the formation or synthesis of ATP from ATP.